Okay, I want to welcome you to our keynote. Statistician Kylie Bemis is a lecturer and a teaching faculty at Northeastern University at the Corey College of Computer Sciences. Her research focuses on the application of large-scale statistical computing for mass spectrometry, and she's developed the bioconductor packages Cardinal and Matter. Cardinal won the 2015 John Chambers Statistical Software Award. She is a tribally enrolled member of the Zuni Pueblo and is active in outreach in Native American and LGBTQ communities, and she's a published author of both fiction and poetry. Welcome, Kylie.
Thank you, Aidan. Hello, everyone. Thank you for joining us. So first things first, I'd like to thank Aidan for inviting me to speak with you and everyone on the organizing committee for putting this together, especially in light of the fact that we can't actually be meeting together in person. So I think it's really great that we could at least continue on with the virtual meeting like this. Okay, so as Aidan introduced me, I'm Kylie Bemis, and I'm going to be talking to you today about the Matter package for out-of-memory computing. So what we're going to do is first I'm going to kind of explain to you what the goals of the Matter package actually are before moving back a little bit and explaining why we developed this package in the first place when there are other similar options for out-of-memory computing, walk you through some examples of how to use it and when you might want to use it, and then give you an idea of some future directions we're going with Matter. So first, Matter is a package for working with file-based data structures. So when you're working with data sets that might be larger than you can fit into computer memory. And one of the goals of Matter is to essentially provide drop-in replacements for your familiar R data structures, things like vectors, matrices, arrays, data frames, and lists, anything that you might represent using these very basic R data structures. We want to provide you with a Matter version of these that you can access just like you would access an ordinary R matrix. But the idea being that the Matter version of these, the actual data might just exist in a file and is only pulled into memory when you actually need it, so that you can work with data sets that might be larger than the memory on your computer. One of the other goals of Matter is that we wanted this to be very flexible. So I'll be talking about some of the other packages similar to Matter, and one kind of common thing between them is they either have their own file format that you have to convert to, or have fairly strict requirements on what the file should look like to work with those other packages. Conversely, with Matter, something we really wanted was to be very flexible in the kinds of data and file formats that you could work with. So one of our goals with Matter is that you should be able to represent any uncompressed binary data where you actually know what the structure is. Obviously, if you don't actually know the file structure, then we can't do anything about that. But if you know what the file structure of some uncompressed binary data file, you should be able to represent that using Matter as a matrix, data frame, whatever have you. We also want you to be able to represent data structures that might be spread across multiple files. So this is particularly common in bioinformatics and especially mass spectrometry imaging, which is the application that I'll be talking about, where a single experiment, you might have very large individual experimental files, but a single experiment might take up multiple files, and you need to combine all of those files into a single data set, maybe into a single matrix. And one of our ideas with Matter is that you want to be able to represent the data across all of those files, maybe in a single matrix or a single array, without actually having to convert or combine all of those files together, because you already have the data in each of those files. We don't want you to have to go through some unnecessary file conversion if we can avoid it. And then lastly, one of the newer things we're trying out is there is a, not quite new, but a new-ish facility in the R language called Altrep. And you know, normally with packages like matter, DelayedArray, we provide these kinds of data structures as S3 or S4 classes. So you might be able to work with them like an ordinary R data structure, like an ordinary R matrix, but really they're not actually an ordinary R matrix. So if you pass them to some other functions that don't know how to handle these, those functions might fail. Conversely, Altrep is kind of a way to extend the native R matrices, the native R vectors, in a way so that we can pass these data structures to functions and they look exactly like an ordinary native R matrix might look. And so that's something that we're experimenting with now, although I think it will be some time before it's really really where we want Altrep to be, but it's something very excited going forward. So now that I've kind of explained to you the goals of Matter, I'm going to back up a little bit and talk about why we felt the need to develop Matter in the first place. Because obviously there are some other packages that are accomplishing similar goals. Now, when we first started developing Matter several years ago, the whole DelayedArray and HDF5 array packages were kind of in their infancy. So although they're great packages now, they weren't a possibility for us when we first started with Matter. I will be talking about HDF5 array and DelayedArray more later. But at the time that we started working on this, bigmemory and FF were kind of the two packages on CRAN that kind of supported this kind of working with larger than memory data sets via file. And so neither one of these packages quite worked for what we wanted to do. And the primary reason being that we wanted this additional flexibility, why we wanted to be able to support open source file formats without having to convert them to a different kind of file format. And so to explain to you that background, I'm going to take a step back and explain the application where I'm coming from, and that is mass spectrometry imaging experiments. So my primary application that I work in is the developing statistical methods for analysis of mass spectrometry imaging experiments. And if you're unfamiliar with mass spectrometry or mass spectrometry imaging, essentially all mass spectrometry does is it you collect mass spectra, and a mass spectra looks something like one of these, where along the x-axis you have mass to charge ratios that represent different masses of molecules, and then the y-axis is the intensity. So the mass spectra can give us an idea of the relative abundance of different molecules in a sample. And mass spectrometry imaging is using mass spectrometry technology, but in a way so that we're collecting thousands, perhaps tens of thousands of mass spectra across the surface of a tissue. And that is what I'm showing here. So for example, here we have a section of a pig fetus, and I'm zooming in on a section here in the heart and showing some example mass spectra that we might have collected from this heart. So for every location on this tissue sample, we have a different mass spectrum associated with each of those different pixels, essentially. And then once we have those thousands of mass spectra, what we can do is we can focus on a particular mass to charge ratio, essentially focus on a particular molecule, and create a false color image that shows the relative spatial abundance of where the molecules are on that tissue. And so that's what I have here along the bottom. So for example, this particular ion here, this particular molecule is mostly abundant in the heart of this pig fetus. And we can see that there are different molecules represented by these different mass to charge ratios that are located in different locations in this particular tissue. And so this is a very exciting technology, often used for proteomics, lipidomics, and metabolomics. That has a lot of exciting applications, particularly in areas like cancer research and drug development. And it produces these very complicated data sets because the resulting data structure is essentially this data cube where we have this mass to charge ratio and intensity axis, but we also have this spatial component, these X and Y coordinates for all of these different pixels. So it's a very complicated data set to be working with. And my first bioconductor package back when I was working on my PhD at Purdue University was developing Cardinal. And so Cardinal is a bioconductor package for the analysis of mass spectrometry imaging experiments. I ended up needing to develop Cardinal because at the time, there wasn't really any R packages specifically focused on mass spectrometry imaging. Now, of course, we have some great packages like MSN-base and all of the packages associated with MSN-base for working with more traditional mass spectrometry. But imaging is really a kind of a very different way of working with mass spectrometry data. So we needed to develop a package specifically for mass spectrometry imaging, and that was Cardinal, which has been very successful. In 2015, it won the John M. Chambers Statistical Software Award, which we're very proud of. And so Cardinal has many features for working with mass spec imaging data. We need to do things such as having spectral and image processing. So being able to smooth the mass spectra, remove baselines, pick peaks of the mass spectra, something that you don't have to worry about in traditional proteomics and traditional mass spectrometry is image processing. With mass spec imaging, we also have to worry about image processing, so doing contrast enhancement or smoothing of the molecular ion images themselves.
There's one question in the poll.
Yeah.
You said to interrupt.
You're muted, Aideen.
Sorry. Can you elaborate on the idea of a data cube? Is it a three-dimensional array?
Yes. So when I say this is a data cube, it can be thought of as a three-dimensional array. Often we don't actually represent the data structure that way, but that's a traditional way that it could be represented. So those three dimensions being, you have the MZ axis. So these are the different mass to charge ratios. And then you also have an X axis and a Y axis. So those are the three dimensions of the array, your X, Y, and MZ dimensions. And then the values of the array would then be the intensities, where those intensities are representing the relative abundance of the different analytes in the sample. For various reasons, we don't always represent it that way, mostly because, as you can see, the actual tissues are usually not a perfect rectangle. Depending on how the data was collected, sometimes we have data for the whole rectangle, in which case it could be represented as a perfect 3D array as a cube, but very often we don't collect data, or we crop out the data in the background around the tissue. So it becomes more of a sparse format where we don't have data for the entire cube. But yes, it can be represented as a 3D array with X, Y, and MZ dimensions. And then we also have, I'll be showing a little bit later, there is a 3D imaging version of this. That is done when you take multiple consecutive tissue sections and kind of recombine them back together to create a 3D image. And in that case, you can have a four-dimensional data cube where you have X, Y, and Z dimensions, and then the MZ dimension as well. Hopefully that answers that question. So again, in Cardinal, we have all of your standard features for a full workflow, including spectral and image processing, visualization tools. So here you can see an image from the namesake dataset of our package. This is a Cardinal a painting of a cardinal on a slide. This was collected by the R. Graham Cook's lab at Purdue. They were kind of showing off the technology in a really neat way. So this was an oil painting of a cardinal, and you can see that we reconstructed what the actual painting should look like from the individual ion images. And then, of course, our focus is statistical analysis. So this is just one example of the kind of statistical analysis that's available in Cardinal. One of which is spatial shrunken centroids, which is essentially spatial segmentation that also performs feature selection. And so this spatial shrunken centroids method is related to nearest shrunken centroids by Tetrani and Hasty, while also incorporating a spatial smoothing component. So you can see the goal here is to segment the image into regions of relatively homogeneous chemical composition. And you can see in this case, what that does is that recovers the tissue morphology. So here, this blue segment, this dark blue is the liver, this red is the heart, and this kind of pink magenta is the brain and some parts of the spinal cord. And then one of the goals in addition to this segmentation is that we also do feature selection. So we get these T statistics that indicate the relative importance of the different ions to these different segments. So kind of the problem that we were running into with Cardinal is, of course, the technology was continuing to advance in mass spectrometry imaging. When we first developed Cardinal, a lot of the data sets were fairly manageable and relatively small. I could easily load them into memory. But very quickly, the technology was getting away from us with instrumentation improvements and high resolution mass spectrometry. And so the file sizes quickly ballooned from, say, a few hundred megabytes or a gigabyte or so up to tens or even hundreds of gigabytes. And this is kind of the issue we were facing with our original version of Cardinal, because we were not really equipped at the time to handle such large files. And kind of the workflow that we go through in terms of analyzing mass spectrometry data is, out of the instrument itself, you get these proprietary raw files. These are often in some sort of proprietary file format. Each different mass spectrometer vendor will have its own proprietary format, and these can be tens or hundreds of gigabytes. And then there was a open source format called IMCML that was developed around the same time as Cardinal, and this format IMCML was an extension of the popular MZML format for exchange of traditional mass spectrometry data, just extended for imaging. And we were really excited about IMZML because it gave us a way to work with data from any vendor. All that the experimentalists needed to do was convert from the proprietary vendor-specific raw files to this open source IMZML format, and we could rely on either the vendors or other people to develop the converters to IMZML. And so that was really exciting. We essentially just needed to support importing IMZML, and that took care of all of the vendors. Now, going from IMZML, after importing that into Cardinal, we then want to pre-process and peak pick the data, and then eventually do statistical analysis. So this is where we run into an issue, because if we can't actually load the data from the potentially very large IMZML files, we can't do any pre-processing or peak picking, and we can't do any statistical analysis. In addition to this, because we have to do this file conversion from the proprietary raw files to the open source IMZML format, we didn't really want to provide a solution that asked all of our users to, okay, now that you've converted to IMZML, we then want you to convert to yet another format so that you can load this data, so you can work with this data in Cardinal. We didn't want to ask people to have to do another additional file conversion after this original file conversion, especially when we were just going to do some pre-processing and very likely reduce the data down to something that's much more manageable in memory. So ideally what we wanted to do was we wanted to develop a way of working with these very large, larger than memory data files without doing some sort of additional file conversion. And that was kind of the main impetus for us to start developing matter. So a little bit of background on what this IMCML format actually looks like, because it's kind of what inspired us to create this flexible way of working with any kind of uncompressed binary data. So IMCML is actually a combination of two files. We have an XML metadata part. So the XML is plain text, human readable format. And essentially it includes all of the experimental metadata. So what kind of instrument was this? What was the lab? As well as metadata on where to find the appropriate mass spectra in the binary file. So we have an XML file and a binary file. And the XML file tells us where we can find the mass spectra for different pixel locations inside the binary file. And there are two kinds of IMCML files that we had to work with. We had a continuous style format and a processed style format. And these were kind of similar, but also different file formats. And we needed something that could work with both of these. So the continuous format is fairly straightforward. This assumes that all of the mass spectra has the same MZ axis. So all the mass spectra have the same MZ values. And so the continuous format, the binary file begins with an MZ array, so the list of all of the MZ values, and then it just has all of the intensity arrays, so all of the intensities for all of the mass spectra. The processed format is a little bit more complicated, and this does not make the assumption that every mass spectra shares the same MZ axis. So this is essentially a more sparse format for sparser data. Suppose we don't want to collect data where the intensity is just 0, where the mass spectrum is just flat. We can store this in this processed IMCML format. And in this case, each mass spectrum is allowed to have its own MZ array. So we have the MZ array for a particular spectrum, followed by its intensity array, and then the MZ array for the next spectrum, intensity array for that spectrum, and so forth. And something we wanted to do with matter was we wanted to be able to represent both of these kinds of IMCML formats in a general way. And since we were already doing the work to try to represent both of these kinds of formats, I wanted something that would be extensible beyond just IMCML, something that might be generally useful to other people as well as ourselves if this format evolved in the future to look a little bit different than it does now. So kind of the central concept of the matter package and how we represent data is in this idea of atoms. So in R, we have this idea of atomic vectors, where an atomic vector is something like an integer vector or a numeric vector, where all of the elements of that vector are contiguous in memory and of the same data type. And so the atoms in matter are essentially the same thing, except applied within a file. So an atom in matter is any contiguous sequence of data elements that lives somewhere in a file. The only thing that really matters is that all of the elements of an atom are a contiguous sequence. So an atom is defined by a data source. So that's often going to be a file. So for example, a file path, so that we know where this atom lives, what file this atom lives in. A group, this is what we use to distinguish, say, between different rows or columns of a matrix or different list elements. Some kind of byte offset from the beginning of the data source. So an atom could live anywhere inside of a file. So for example, this atom on the left here, I have my storage, I have some sort of file living on my storage, and then inside this file, I have an atom, and I can see this atom doesn't start at the very beginning of the file, but somewhere inside the file. And so each atom is defined by some sort of offset from the beginning of the file or the beginning of the data source. And then as well, an extent. So this is just the number of data elements in the atom. And each atom is just every element is a single data type, just like an atomic vector in R. Some additional characteristics of these atoms are there's an index offset and an index extent. So these are essentially the indices of the elements within some sort of parent matter object. So if I can have a vector that is made up of multiple atoms, and these index offset and index extent define where within that vector the atom lives. I'm going to show you some examples of this in a moment. But the idea behind an atom is because an atom is a contiguous sequence of data elements, we can always load an atom into memory in a single read operation. So then the idea behind these matter data structures is that we can build a data structure up from these individual atoms. So we have atoms that come from different locations in different files, potentially multiple files. And the idea is a matter object is essentially just a collection of atoms. So it's essentially just a collection of pointers to different locations within different files. And the idea behind matter is that it kind of abstracts this structure away from the user so that when you're using a matter object, you don't have to care what these atoms are, where they're located in these different files. All you see is what looks like a regular vector or a regular matrix, and matter takes care of rearranging all of this data, grabbing it from the files and rearranging it so that it looks like a contiguous vector or a contiguous matrix in R. So we can treat this like a single data structure, for example, a vector, with atoms that can come from any number of different files, any number of different file locations. And then this data is only loaded into memory whenever you actually request it. So as an example of how we can use this, so here I just have an example of creating a very small matter vector in the matter package. So this is an example where I've already loaded up the matter package, and I can just use the matter function to create a simple matter vector. By default, I'm not specifying any file location here, so it's just going to create a temporary file in your default temporary directory. And this will create a temporary file and write this integer vector 1 through 10 to this temporary file. Now, X is a matter vector that happens to contain just a single atom, and that atom happens to point to the entirety of this one particular file. And if I look at the atoms in this, I can see I just have one atom of data mode int. The offset is 0 because it starts at the beginning of this file, and it has 10 elements to it. So here I'm going to create a second matter vector in the exact same way, but now I'm going to do 11 through 20. And again, this will create a new temporary file. I could specify a different file location if I wanted, but this will by default just create a new temporary file. And this file too is going to be a new file. And what I can do now is, now that I have these two different vectors, I can combine them together into a new vector that combines both of these atoms from these two different files. And that just works seamlessly, transparently, without the user having to think about the fact that these two atoms, these two original vectors, are coming from two separate files. I can index it just like I could in an ordinary R vector. And I can see if I looked at the atom data for this new Z matter vector, I can see it has two atoms that come from two different files, each with 10 data elements. And so it's really simple and really cool. Here I can even combine these using rbind into a matrix. This one becomes a little bit more complicated now because now I have a matrix with 4 atoms, and each of these different atoms points to a different file. If I wanted to do something a little bit more customized, in the last couple slides, I was just creating a vector and allowing it to create a temporary file, but I can also specify where the files are, as well as the specific offsets and extents if I want to create support kind of a custom file structure. And this is essentially what we do when we're reading IMCML in Cardinal, is we're doing this kind of custom matter data structure where we can specify all of the offsets and extents for each of the individual atoms, which then correspond to the different mass spectra in the IMCML binary files. Something that's really useful that comes out of this is matter is useful if you just happen to have some kind of binary file that you want to read, even if it's not necessarily larger than memory. So this is an example from an Analyze 7.5 header file. Analyze is an MRI data file format. It also happens to be used for mass spectrometry imaging. But this is just an example of kind of a common binary header file where it's represented as a C struct. So you have these different data fields that are these different kind of C data types, ints, characters, shorts, and so forth. And in Matter, we have a shortcut function called struct that essentially takes care of representing this kind of C-style struct so that you can very easily represent these kinds of header files or whatever kind of files that you might need to represent. And so again, this is a header file that's going to be quite relatively small. It wouldn't be a problem reading this all into memory, but Matter gives us a very nice and neat way to represent the file structure of this header file so that it's very easy to read. And then we can access the different fields of this header file just like we would a list. We support delayed operations, much like DelayedArray. So here, for example, if I take the log of a vector, the log will be applied only when I actually access the data. So for example, doing +1 and taking the log of a matter vector, this will not actually touch any of the original data, but it will just apply this kind of operation on the fly whenever I try to access the data. And all of this happens at the C++ level. Here's just an example of doing some linear regression. So this is just a toy data set, 1.2 gigabytes, relatively small, but just showing that we can do a linear regression using the big LM package to fit a linear model on this matter matrix without ever loading it into memory all at once. And something really exciting going forward is we have this Altrep feature of the R language that is relatively new. And this is something we've recently built in support in Matter, is you can specify that when you coerce Matter objects to their native R equivalents, you can specify that you want an Altrep version of these. And so that means when I do as.matrix on a Matter matrix, it'll give me what looks like an ordinary R matrix, but underneath at the C level, there is actually a matter data structure backing up this what looks like an ordinary R matrix. And what that means is we could pass this to essentially any R function, and it'll act just like an ordinary R matrix, even if that function doesn't know what to do with a matter specific data structure. There are some challenges associated with this because of the way Altrep is implemented. Sometimes if the code is not aware of Altrep, it'll end up loading everything into memory anyway. So that's an area for future improvement. Here's an example of us taking the iris data set, turning it into a matter data frame, and then turning this data frame into an Altrep data frame. And kind of the lingering question here for future work is often Altrep, if the code is unaware that it's an Altrep object, many times the data might get materialized into memory anyway. So here's just an example of timing, accessing a matter vector versus the Altrep. And you can see that sometimes the Altrep is a bit slower, and this might be due to some ongoing development limitations in how Altrep is implemented. So the package landscape, this is filling in what I showed you earlier. So we have packages like bigmemory and FF. These are kind of older packages on CRAN for working with large file-based data structures. And newer, we have matter and HDF5 array. And these are just kind of the different features. Matter, one of the main things that we try to emphasize is that we have this flexible structure so that you can kind of support custom data structures. We support many different kinds of data representations at the R level, including vectors, matrices, data frames, and lists, which some of these other packages do not. And just kind of a quick shootout in terms of speed. So here, all I did was a very simple calculate the variance for all of the columns of a 1.2 gigabyte simulated data set. This was using just a simple v-apply here. I didn't want to use any of the optimizations so that there was no particular advantage to any particular package here in terms of, say, delayed matrix stats or something like that. So certainly all of these speeds could be improved with package-specific optimizations. But this is just to show that matter tends to be quite fast. It's faster than FF and HDF5 typically. Bigmemory here was the fastest, but that's also because it tends to keep more of the data in memory. And I'm sure there's probably ways to further optimize the HDF5 file for this test, but this was about as good as I could get it for now. So these are just some examples of using matter to analyze some relatively large IMCML files. So here's an example, not too large, but this is a 26.48 gigabyte 3D mouse pancreas experiment. This was done on a laptop with just 16 gigabytes of memory. So this is an example of that kind of 3D mass spectrometry imaging where they took multiple tissue sections and then we can kind of reconstruct them into a 3D image. This is the same data set where I've calculated the mean spectrum as well as the TIC, the total ion current for all of these pixels, just visualizing these. And in our paper on matter back in 2017, we applied principal components analysis to all of the continuous IMCML datasets in a GigaScience repository of 3D MSI data. And matter outperformed bigmemory. This was before HDF5 array was really a thing, so that's why it's not included here. And FF could not really do anything here because there's actually a 32-bit limit on the file sizes. So the last thing I want to mention is, so we have these continuous IMCML formats and also this processed IMCML formats. And the processed IMCML, as I mentioned, is kind of this sparse format. So how do we actually represent this kind of sparse matrix structure in matter? And this is something that's really unique to matter. So this is a smaller data set. This is just 850 megabytes, so it's not large, but it is a sparse processed IMCML file. So the way, if I look at the spectra, I can see it's very sparse. So here, about 2.13% density. I can see most of these are zeros. And essentially what we're doing to represent these sparse processed IMCML matrices are, we can basically store the matrix elements as key value pairs. So these processed IMCML, each mass spectrum has the MZ values and the intensities. And essentially what we're doing is, rather than storing the row ID or the column ID, the idea behind the sparse matrices in matter is that you can use any appropriate value as kind of the key to represent the row or column of a particular data element. So here we can use the MZ values to represent the row in this sparse matrix. Now, one thing we have to take care of with this is that there is some error associated with these MZ measurements. So they might not be exactly the same, even when they might correspond to what should be the same value. What this means is our sparse matrices actually have to bin data on the fly. So these are sparse matrices that are file-based, allow you to use any appropriate value as the row or column ID, and also can bin data on the fly when you read them into memory. So for example, here we have two mass spectra. These are the MZ values and intensities. And when I read these mass spectra into memory as the columns of a sparse matrix, some of these values may get binned together depending on what I have as my kind of canonical list of MZ values. Because so, for example, 404.1019, 404.1015, these might get binned together depending on the tolerance or resolution that I have set. And this is something that's really nice when you're working with mass spectrometry data in this kind of sparse format. So in conclusion, when would you want to use matter and where would you want to use it? Whenever you're doing any sort of I/O with open source binary files, so as I showed here, working with IMCML, I showed an example of this analyze header file. So anytime that you're working with a binary file where you know what the file structure is, and you don't necessarily want to convert that into something else like HDF5, then matter is really useful to use. And this is true even if the file might not be larger than memory, as I showed with the analyze file. This can be a very useful way just to take care of file I/O. But it's very, very speed and memory efficient when working with larger than memory files still. Also, if you happen to want to write or use some kind of user-defined custom file format, or just whenever you want the speed and memory efficiency of these kind of essentially very simple flat files, HDF5 has a very nice and rich feature set that can be more flexible in terms of a file format. So you have these different chunk sizes and shapes to these different chunks and compression that you can deal with HDF5 that isn't possible with matter. So those are things that are very nice with HDF5, but can also affect the speed and efficiency of working with it. And then also, as I showed in the last couple slides, we have these new file-backed sparse matrices that we're using to work with this processed sparse mass spectra data. And this is something that we want to try to expand support with in the future. So in terms of future directions, we want to further experiment with these Altrep objects. I have some colleagues that are trying to investigate some alternatives to Altrep for representing native R objects as well. These are called, they're calling these UFOs, which is an exciting thing. I'll mention the people working on that in a moment. We're working on providing a delayed array realization backend using matter. So you can certainly put matter into a delayed array right now. That'll work out of the box, but we're working on creating a package so that it's easier to use delayed array with matter. So it's perfectly possible to use delayed array and matter together with matter as the backend. We also want to expose more of matter at the C++ level. So we're looking at using the beachmat package, for example, for that. And then we're also going to be exploring further improvements and support for these file-backed sparse matrices that we've been developing. Lastly, I want to do a quick advertisement of another package from our group, MSStats. So if you're doing traditional mass spectrometry, proteomics, after you've worked with the raw data, processed the raw data in something like MSNBase, MSStats is a great package for doing quantitative analysis for mass spectrometry-based proteomics. And these are just a list of features for that. And this is developed by Olga Vitek and Mina Choi. All right, so that concludes my talk. I'd like to, again, thank Aidan for inviting me. These are some of the people who have contributed. So Olga at Northeastern RPI has been extremely helpful, as well as Jan Vitek and Jan Vitek's group in Prague for providing some input in terms of the R language, Altrep, and that kind of thing. And then, of course, Graham Cook's lab at Purdue and Melanie Foyle have been very helpful in terms of helping us understand the mass spectrometry imaging data. Thank you, everyone.
Thank you. Thank you so much. We have a lot of questions. A popular question was about sparse matrices. Does it read and support sparse matrices? And I think probably akin to that, people thinking about sparse matrices that for single cell RNA-seq or spatial single cell data.
Yeah, so currently for these sparse matrices, you can create your own. We don't have out-of-the-box support for things like RNA-seq or things like that, but if you were able to, right now it requires a little bit of knowing how to put them together. So for example, here what is required is that we have each of the keys and values here are represented as matter lists currently. So each element of these lists are vectors where the keys in this case are the MZ values. The values, of course, are the intensities. And then we can apply kind of a canonical list of MZ values that define the rows of this sparse matrix then. And so what you would need to do is you would just need to define what are the keys and what are the values for your sparse matrix. And then you can use that to either create a compressed sparse column or a compressed sparse row matrix once you have those. And there are some examples of that, but currently there's no out-of-the-box support for something like RNA-seq. But this is something we hope to kind of keep working on these sparse matrices. Right now, they do require a bit kind of knowing what you're doing to put them together, though.
And so does it work with the sparse matrix packages then as well?
No. So because what we're doing here is working with file-backed sparse matrices, right now, these are much more limited in how you put them together. This is, again, something we want to work on more in the future. So right now, if you access some part of the sparse matrix, it'll return to you a dense matrix. And that's something that we want to change when we implement some of this delayed array stuff, so that if you access a section of the sparse matrix, it'll return to you a sparse matrix from, say, the matrix package. But right now, although they follow kind of your standard CSE or CSR sparse matrices, they are their own separate thing, but we want to improve the interoperability with the matrix sparse matrix packages.
Cool. Another question is it possible to do filter, select, mutate operations on a matter object like a regular data frame?
So any of these dplyr functions, because in dplyr, they're all implemented in C++ or via a database. So right now we don't have direct support for those, because that would be kind of a major undertaking to duplicate all of that. So that's something I'm not sure if I'll do that myself, or that might be something that someone else might undertake in creating those kinds of dplyr style functions for matter objects. Also, in addition to that, because the data frames in matter are kind of underused and undertested right now, because we've been focusing on matrices as the main thing that we use. And so we want to do some more work with things like data frames in terms of figuring out how useful they are and what operations are most useful on those.
But I'm sorry, just thinking about that question, can you do those operations then on a matrix? Does it work?
Not directly. I mean, you can of course subset the matrices and get a matter matrix back as the subset rather than a dense R matrix. But and there is some support for if you, for example, like the which function. Currently, there is a which function implemented in Matter at the C++ level, and we do have logical operations are delayed as well. So for example, here we have these delayed operations. All of the ops, so arithmetic, comparison, and logic are all supported for this. So although you can't directly do the filter mutate stuff in using the dplyr functions, anything that you could do kind of in vanilla R should work. Again, since things are on file, it might be a bit slower, but yeah.
Cool. So another question kind of related to that is operations on the data frames or matrices. Can you combine multiple smaller data frames or matrices that fit into memory into one giant data frame or matrix that cannot fit into memory? How is it saved and reloaded for later use?
Yes, that's exactly the idea, is that especially with these mass spectrometry imaging data sets, lots of times the individual files might be small enough to fit into memory, but taken all together, all of these different IMCML files might be in aggregate larger than memory. And that was one of the things that we wanted to take care of and be able to support with Matter. And so with Matter matrices, that's very easy. If you just do an R bind or a C bind on matrices of the same, you either have column wise or row wise matrices, and if you do an R bind or C bind on kind of matching matrices like that, then you can create a matrix where the different columns come from different files. And in fact, that's exactly what's happening in this slide, although on a much very small scale. So here, we have kind of these four quadrants of this matrix, and each of these is coming from these two different files. And so that's what we routinely do with these IMCML files, is we have to create these large matrices where different columns come from different files. Yeah. Oh, and to answer the how is that saved, currently none of that is actually saved and rewritten because we want to avoid file conversion whenever necessary. So if you wanted to explicitly write that out, that is something that you could do. But the idea is that you would keep whatever your original files are and avoid the file conversion. And essentially all you need to do is just rebuild these Matter matrices very simply anytime you want to work with that data again. The idea being that since you're not actually reading all of that data into memory, it'll just take a few seconds since you're essentially just reading metadata.
We've got a lot of questions. I think we're going to have to put some in Slack to you. There's another one about, can you explain more about Altrep and what is it?
Yes, so I didn't go too much in detail on exactly what Altrep is, but essentially when you have, so if you're working with something like a delayed array or a delayed matrix or a matrix from the matrix package, these are things that you can interact with very similarly to native ordinary R matrices, but they aren't actually ordinary native R matrices. They're implemented as four classes that have, and you interact with them in a slightly different way. And so that means if you pass a delayed matrix or a matrix from the matrix package or whatever, or a Matter matrix to a function that doesn't necessarily know how to work with those objects, that function might fail, especially if that function then calls some sort of C code or C++ code. Because then especially at the C and C++ level, that code will not know what to do with, will will often not know what to do with something that's not an ordinary R matrix unless it was specifically designed otherwise. The purpose of Altrep is a feature, a new-ish feature of the R language, is that rather than creating new S4 classes that look and behave similarly to matrices and vectors, what it's actually doing is going in and replacing the data pointer of an ordinary R matrix at the C level, so that even if you have something that looks and behaves exactly like a native ordinary R matrix everywhere. And if you pass this Altrep matrix to C code, even at the C code level, if it asks for some data from the data pointer or some window of data, this Altrep object will behave like an ordinary R matrix, but it will get and retrieve that data from, in this case, a matter vector. And the idea being that you can pass these Altrep objects into C code that may not be aware of what the underlying data structure actually is, and it's able to behave like an ordinary R matrix, so the C function can get away with doing whatever it wants to it. Again, the drawback to that being that sometimes the data often can get materialized in memory anyway, since the C code often still has to have an idea that this is an Altrep object. And there are some issues with efficiency as well that hopefully we can figure out down the line.
Very cool. I think we're actually at time. And I think we'll post the rest of the questions in Slack for you. I want to thank you again. It's been a really great talk and I've learned lots. Thank you.
Thank you.
Thanks so much, Kylie.